on the street is that the replication worksheet is less challenging than others. All right, so today we are going to move into our next part of our central dogma. So we're going to move into transcription. There is a worksheet posted for transcription. Just wanted to check if I had any errors on here that I needed to. Okay. We did correct. All right. People apparently find me scary, so I don't know. That's what I heard. I really hope that's not true. I try not to be scared. Okay. Okay. Makes me feel better. I've lost the page of my work. What's going on here? All right, there we go. Okay, so on chapter 10's worksheet, I don't see that I made any corrections while I was writing the answer key. So I think you should be good to go on that. That is due next Friday at midnight. Should you feel like working ahead, there are also worksheets posted through chapter 11. So feel free. You guys are ready to power through these chapters. Okay, so don't forget that the replication or chapter 9 worksheet is due tonight at midnight. Yes?
Yes.
Okay, so I can give a few minutes towards the end of class to go over those if you have questions on them. So I'm happy to do that. Otherwise, the next thing for this class will be exam next Friday. Exam on Friday. So that again will be on DNA structure, DNA replication, and DNA transcription and RNA processing. I was looking for a good introductory overview video for transcription today, and I ended up in another wormhole of weird videos. But I came across one that was from MIT. So they have these online courses that you can take, and one of them is the fundamentals of biology class. But it's a faculty going through transcription, but he must have 10 chalkboards behind him. Have you seen those old school chalkboards that you can roll up and down? He had those, and he had drawn up his entire lecture out. I was in awe. It was kind of amazing. That's what I did with my morning. Okay, so I feel like there was something else I need to tell you, but I can't remember now. So what we're going to do today is I just want to do the overview of transcription. So talk about sort of the process as a whole, the components that we need, and the things that are involved. And then on Monday, we'll come back and we'll start getting into the more specific details of the initiation, the elongation, and termination. So we'll talk about the 3 stages today, sort of briefly or generally. I will talk about promoter sequences, and then sort of the other pieces on here are going to be more specific to next week. So to start with, I want to go back to our central dogma and remind you that this is what we are walking through right now. We have now done the first piece, our replication loop up top. What I want us to think about for a few minutes now is we are going to be looking at how we take a gene coding sequence and from that gene coding sequence get to our end product. So in these figures, we almost always show our end product as being an amino acid chain or a protein. But I think that it is probably useful at this point to have a good understanding and a good definition of what a gene actually is. Okay? So take a minute, work with a neighbor or two, and come up with a good definition for gene. So when I'm talking about genes, what am I talking about? What am I saying? So come up, define gene amongst yourselves. Why is everybody acting like this is such a hard hard task.
Yeah. It's a Monday, Friday.
It's a Friday.
But it feels like a Monday.
No, it does not. It feels like a Friday. This whole week felt like a week of Fridays.
I don't think we've been in the same week.
Lowercase G, not like the name.
That's how we identify.
Thank you, brother.
There's like 6 subdivisions in the chapter.
That's a good start. All genes are located on chromosomes. It's true. You guys look ahead in your notes, like I know this time I did not put this in here. Is it on the next slide? Is that what I do all the time? Do we have definitions? Nobody wants to. All right. How have we defined a gene? Who would like to share their definition? No one. Is that because we couldn't define it? Or because we just don't want to talk? That one. Cool. All right. Well, somebody give me an answer. I'm just going to call a name. I'm gonna go get my popsicle sticks. I have popsicle sticks with numbers written on them, and then the numbers correspond to the numbers of everybody's name on my list. I actually started that way, and then I was like, I'm gonna have to get new sticks every semester. So I just did yours. Yes? All right. We're gonna do it then. And, oh, it's Hunter who's not here. Again. It is Grayson. Grayson, would you like to share with us your definition of a gene? Do you have any characteristic of a gene?
Nucleotides.
Okay. A nucleotide what?
That specifies an amino acid.
All right. I'm going to add a word to that. It's a nucleotide sequence, right? And then you said that specifies an amino acid. Okay. That. Is this an all-encompassing definition of gene? No. Okay, so what's missing? Or how can we change it to be representative of all genes? Okay, so you would be saying then a nucleotide sequence that specifies a protein.
Like a protein that codes for the protein. Like the amino
Okay.
acid chain makes up the protein.
Okay, yes. So, okay, sure. Okay, so we've added that we're getting an amino acid sequence that results in a protein that causes a trait. Okay? That's true. Are there any other types of genes? Or any other things that genes might do?
They are inherited.
They are inherited. An inherited DNA nucleotide sequence. Did anybody else come up with something additional that genes may do?
They include the coding regions.
Okay, so they are a nucleotide sequence that codes for a specific amino acid. Yeah. So an inherited nucleotide sequence coding sequence. An inherited DNA nucleotide coding sequence that specifies an amino acid. Do all genes produce amino acid sequences? What else might DNA sequences code for?
Traits.
Traits. So traits are going to be a result of proteins which come from our amino acids. Say that again?
They might not necessarily show it. Transcribed.
Okay. Phenotypes are still trait driven or trait based? Yes.
RNA translation.
You said the right word, but then you added an extra word.
RNA.
Yes. So we have genes that are inherited, that are nucleotide coding sequences, that specify RNA sequences. So in addition to amino acids, which this is how we often think of a gene, is that we have a DNA sequence that gets transcribed and translated and becomes a protein. That's how we talk about our central dogma. But there are other products of genes that are not amino acids. So this definition is very specific to proteins and to amino acids, and that's fine. This is how we do typically define a gene. But we could actually make this much broader and say that specifies a, draw a line because this isn't wrong, but it's not quite complete, a functional product. So in our genome, we have regions of DNA that are going to result, so when they are transcribed, they are going to result in usually RNA sequences. So we have, right here, all these different types of RNA. We have ribosomal RNA. Is that a functional product? Do we use it in our cells for something? Yeah, without our RNA, we can't go through this process. We can't actually get an amino acid. But our RNA sequences come from our DNA. Same thing with our transfer RNAs. Again, without transfer RNAs, we can't actually make a protein. But that is the functional product of DNA as well. So there is a gene. Where are our tRNA sequences found? That's where tRNAs are located. That's not where the genes are. Mitochondria, 22 tRNA genes are from our mitochondrial genome. And then we have all these other RNAs, which you probably maybe haven't heard of. These are much more specific. but we have SNO and SNRPs and mini-RNA, microRNAs, siRNAs. All of these, again, are functional. They have important roles in our cells, but they are not amino acid sequences, they are not proteins, and they do not code for specific traits. Okay? So usually when we define a gene, or when we're defining a gene, I think that we need to be sort of specific in what we're actually talking about. So for our definition, we're going to be pretty broad, and we are going to call it a segment of DNA that codes for or results in the production of a functional product. With the understanding that a functional product is often a protein or a polypeptide chain, but can also be an RNA sequence that then functions in other cellular processes. Everybody good with that? All right, so for transcription and translation, we are going to still be referring to our DNA, especially in the transcription process, but we're going to focus really heavily on RNA because we have lots of different types of RNA that are going to be involved in transcription and translation, primarily mRNA, which is our messenger RNA, rRNA, which is our ribosomal RNAs, and tRNAs, which are our transfer RNAs. Those are all going to be part of this process. So, since we're going to be talking so much about RNA, I figured that we should come back to its structure, since we only sort of briefly talked about RNA structure when we were doing our DNA structure lecture a few weeks ago. But I think we know most of these things. So here is an image of an RNA molecule. We already know at this point that both RNA and DNA are polymers. So they are both comprised of monomers, and the monomers are essentially the same between the two of them. Those monomers are nucleotides. Both RNA and DNA are made of nucleotides. The types of bonds that we see in our backbones are the same. So we still have our phosphodiester bonds that hold together our phosphate groups and our sugars. Major difference between DNA and RNA is the type of sugar that we have. In DNA we have deoxyribose, and in RNA we just have ribose. The main difference between these two types of sugars is whether or not we have a hydroxyl group on that 2' carbon. So in DNA, there is no OH group right here. That makes it a deoxyribose sugar, right? Because we've lost an oxygen essentially, deoxy. Whereas in our ribose sugars, we do have that OH group coming off that 2' carbon. And actually, if we want to get really picky, we would call this 2' deoxyribose. But most people do not do that. The nucleotide bases are mostly the same between DNA and RNA, with the exception, as we know, of uracil for thymine. Again, structurally very similar. The difference is a methyl group on, I think there's a methyl group on uracil. Maybe it's on thymine. Anyways, there's a methyl group in one of them and not in the other. We know RNA is single-stranded. So is RNA more or less stable than DNA?
Less stable.
Less stable. One of the primary reasons is because RNA is single-stranded, so it doesn't get protected and it breaks down super easy. In DNA, our secondary structure is simply that double helix, so we get two linear DNA molecules bound together and then they spiral up. In RNA, we have really interesting and complex secondary structures, so I know that that's a little bit hard to see, but next chapter when we're talking about our transfer RNA, we'll be talking very specifically about secondary structure in RNA. So we have, this is all one single strand of RNA, but it has folded in on itself because we have attraction or complementary nucleotide sequences within that single strand, and so they're drawn to each other and they form those hydrogen bonds. So we do get sort of these interesting loops and hairpin sort of turns in our RNA. In some of our RNA molecules, that structure is very important. So we'll see again with our tRNAs, all of our tRNAs have a specific stem and loop structure that is essential to their overall function. All right, so that's our RNA. Again, these are the different types of RNA that we have in our cells. We will focus really heavily on these first three, but we may mention some of these others. You may notice on here when you look at the different types of organisms that have these different RNAs, that again our prokaryotic friends are much more simplistic. They have rRNA, mRNA, tRNA, and CRISPR RNA. Eukaryotes become much more complex, and they add in SNO and SNRPs and mini RNAs, or sorry, microRNAs. SNRPs is like my favorite. Kiwi interacting RNAs. Right? These are all sort of upper molecular mechanisms that we won't get too deep into. I will say, however, has everybody heard of CRISPR? CRISPR-Cas9? What is CRISPR? Not the RNA specifically, but what do we know that term in reference to? Where do we hear? Yeah.
Genetic modification.
Yeah. So CRISPR and Cas9 are some of the technologies that we're using for modifying or manipulating genomes within cells. So I don't know if I wrote this, but you can buy a CRISPR-Cas9 set and try and manipulate your own genome. But we are actually using it for like medical purposes. So we are doing gene editing therapies. We are trying to remove mutations in nucleotides and gene sequences that are disease causing. And there has been some good success. There's still a lot of ethical pieces that we're trying to work out in terms of how far should we go, what should we do, who should we be doing this on. But that's some of we're using these CRISPR RNAs in order to do that because they can be used to target specific DNA sequences and replace them with correct or non-modified, non-mutated sequences. Okay, so overall, our transcription itself is going to synthesize DNA templates, but it is fairly selective in what it does. So when we are transcribing something, we are going to be transcribing a specific region of DNA. So you guys talked before about like upregulating and downregulating gene expression. I did recently talk to Dr. Hammond and Dr. Urban about what things you learned in classes with them, and I was told that you have seen these things before. Maybe not specifically about upregulation, downregulation, but you've definitely seen transcription and translation before. Yes. Kylie's the only one willing to admit it.
I've seen it, but I don't remember.
That's fair. Okay, so transcription is going to lead us to this point eventually, right? So if we are transcribing a particular gene and we're making many RNA copies, those RNA copies will then be translated into amino acids, which fold into our proteins, which then dictate our traits. Does everybody agree with that? Okay. So are all of our genes transcribed and translated and expressed all the time? No. And so I think we started talking about this sort of at the beginning of class where we said every single one of our cells has the same genome and all of our genes are present in every single one of our cells. But each of our cells expresses a different set of genes so that they function as part of a specific organ. Great. So, when we're saying that transcription is a selective process, what that means is that each cell is going to transcribe specific genes at specific times, and that we can turn off and on or regulate how many mRNA copies of a particular gene we get, depending on what the cell needs at that particular moment. All right, so let's just walk through the components of transcription. These may look kind of similar to us or familiar to us. First, we need a DNA template. That DNA template is going to be the gene, or it's going to contain the gene sequence for whatever our functional product is. So if we are a kidney cell and we need to pull more water out of our urine and put it back into our bloodstream, we may find the DNA template for aquaporin production. Aquaporins are channels that allow us to remove excess fluid from waste urine and return it to the body. So we'll find that gene that codes for that functional product. We will then take our raw materials, and our raw materials are sort of the things that we're going to need to build the RNA, mRNA, and then eventually the protein to make that functional product. What was our raw material or our required pieces for replication? We still have a DNA template to start with. What do we need to build a new piece of DNA? We also use them in lab this week. D and TPs. So D and TPs are going to be the real raw product that we need to make a new strand of DNA. And in transcription, we're making an mRNA. So our raw materials are going to be what we need to make an RNA. And that would be ribonucleotide triphosphates. Right? So if we're making DNA, we're going to use deoxyribo trinucleoside triphosphates. If we're making RNA, we're going to use ribonucleotide triphosphates. And then we need the transcription apparatus. So again, if we're doing replication, we would need all of those proteins and enzymes that come in, find that's the origin of replication, and progress that forward. With transcription, we are also going to need a series of proteins and enzymes that are going to come in and identify the start of transcription, so our promoter region, copy the DNA to mRNA, and then end the process. And so these actually are a couple of images that sort of show transcription and process. This is the hand-drawn one. But here is the DNA template. Coming off of the DNA template, all of these pieces that make it look leaf-like, those are actually mRNA transcripts. Right? And so we can transcribe multiple regions along a piece of DNA at one time because we may have multiple genes located, or we may need tons of mRNA copies. So we may have multiple RNA polymerases moving down and making transcripts sort of one after the other. And so this is sort of what it looks like in real life. And so here is sort of the overview image of what this process looks like. What does this enzyme remind you of? DNA polymerase, which we use to make DNA. This is RNA polymerase because we're using this to build an RNA polymer. So they are somewhat similar in their structure and function. but they do have different end results. Let's do that in a little bit. Just a more simplistic overview of what's happening. So in transcription, we start with our DNA template. We find our region of interest, and so on this region, at the beginning, so somewhere in here, so in here, we're going to have a promoter. Right, that is a specific DNA sequence that the transcription apparatus, so all those proteins and enzymes that get things started, recognize and can sit down on. At this end, we will have a terminator. Which is another DNA sequence. When the apparatus works its way down our template DNA strand and hits that terminator sequence, it essentially disassembles and lets go of everything. That is the end of the process. Both the promoter and the terminator are DNA sequences, so they're already built in sort of before and after the actual gene. Just like our DNA polymerase, our RNA polymerase is going to build a new molecule in the 5' to 3' direction. So in this case, our RNA polymerase would sit down at this end and copy this as the template strand, because it is the 3' to 5' direction, which means it can build an mRNA molecule in the 5' to 3' direction. With that being said, both strands of DNA can act as both template and non-template strands. So the template strand of DNA is going to be the strand of DNA that we are actually copying into mRNA sequence. You may also hear it called the coding strand. The non-template strand is going to be complementary to the template strand. So, if we have a sequence of DNA,